Good afternoon, everyone. First, I would like to thank the OIS organization for giving me this opportunity to share with you today a little bit of what we're doing at InGel. So InGel Therapeutics is a preclinical company leveraging stem cell therapies and biomaterials using regenerative medicine to treat retinal diseases. But before I tell you about the technology, why did we create InGel? Well, the reason why is that we've seen many scientific and clinical learnings in stem cell therapy in the back of the eye, but limited commercial success. And I'm talking here about more neuronal stem cell therapies, such as photoreceptors and retinal ganglion cells. So with our background in cellular science and biomaterials engineering, over the years, we have identified a couple of key reasons why these cell therapies have not been successful, such as the cell purity, the viability, the shear stress, but also the environment in which these stem cells are being injected, which is a diseased eye, very hostile microenvironment with very specific cell-to-cell interaction. So with that in mind, we created a novel technology at InGel to address these key issues. And the first disease we want to target is retinitis pigmentosa, and I'm very lucky to go right after Pierre, so he told you a little bit about RP. But if there's 2 points I want to make right here. The first one is, even though RP is one of the most common inherited retinal disease with very high unmet need, due to about more than 100 different distinct mutations, at InGel, we're targeting a mutation-agnostic mid-stage approach for RP. So this is basically the yellow square that you see on the screen right there. The second point is that means that we're focusing on rescuing and repairing cone photoreceptors. But to do so, we needed to understand why are these cones dying, even though they're not mutated. So we saw a lot of primary drivers: synaptic connection loss, metabolic deficiency, oxidative stress, or even phototransduction malfunction. So to tackle these primary drivers, we created our lead asset, IGT-001. It is an intravitreal stem cell matrix acting as a biofactory to restore endogenous cone function. So within the syringe, we have our novel stem cell, a rod precursor cell that is encapsulated within our matrix made of hyaluronic acid and gelatin, and we perform an intravitreal injection of this stem cell matrix. Then the technology works in 2 specific steps. The first one is that we've designed this matrix so that it adheres onto the inner limiting membrane, offering a targeted delivery even with an IVT injection. And second step, the MOA of the technology, is that this biofactory will deliver a specific secretome that will be transported through the retina in order to target cone photoreceptors to rescue and repair them. And while that happens, the hydrogel fully degrades, the rods are being cleared away, which offers us a potential for redosing for patients. So I've just talked about the secretome. Even though we're a stem cell company, we actually did a lot of work to understand what is this secretome and what is the MOA of IGT-001. And what I can share with you today is that we've identified 10 key proteins that are the primary driving effects of IGT-001, targeting glycolysis with RDCVF, passaging 1, but also the lactate pathway, targeting oxidative stress with lipid peroxidation, or even the NRF2, which is a well-known pathway within the retina. Overall, this early work on potency and release assay for IGT-001 allows us to understand why we're restoring cone homeostasis, which leads to the improvement of phototransduction, synaptic connection, cone fueling, and oxidative stress protection. We've done a robust preclinical development showcasing the MOA, safety, and efficacy of IGT-001 across mutations. We have done work on the 4 major animal models, RD1, RD10, RHO, and P23H. And in the interest of time, I'm just going to share with you a couple of these results that we've done preclinically. The first one is on the RHO model. We were looking at behavioral vision and functional rescue. We measured both contrast sensitivity and visual acuity in these mice. And what you can see is that in blue, you have the IGT-001 group, and in gray, sham or vehicle. And we were able to observe a significant improvement in both contrast sensitivity and visual acuity following the injection of IGT-001. And keep in mind that this is only a single dose treatment right there. But this is more for cone rescue or cone neuroprotection. And we wanted to see if we could take IGT-001 to the next level. And to do so, we actually went a little bit later stage into another model, the P23H mice. We injected at a time where these mice are almost completely blind. Their ERG, as you can see, is almost 0. They basically don't see anything. What we were able to observe is that only 1 week after injection, we were able to rescue or repair almost 50% of the ERG photopic B wave in these mice. And that was confirmed with the anatomical staining we observed at week 7, where we were able to see in the IGT-001 group cones with outer segments and synaptic connection to the inner retina, which was not seen in the sham group. So just taking a step back from science and data right here, I mentioned that InGel with IGT-001 is focusing on a mid-stage approach gene-agnostic treatment for RP, meaning that we are not overlapping with the early gene therapy or cell replacement and optogenetics that was just presented. We are in this mid-stage doing neuronal protection and repair, but we have a unique differentiation. First, we can perform visual recovery beyond rescue, as I just shared with you with the P23H data. Second, we have a broad patient population. We're mutation agnostic, and we have the potential to redose. And finally, once again, even though we are a stem cell therapy company, we have a well-characterized and in-depth understanding of the molecular mechanism of action of IGT-001, also with the use of our proprietary delivery scaffold. The team behind InGel is very passionate, working daily on making this a reality. We're a spin-off from Harvard Medical School, the lab of Michael Young and Myron Spector, and we have amazing clinical advisors that are steering us the right way to make an impact on patients. Just to summarize, we're a Harvard spin-off. We have a technology which is both a stem cell therapy and a biomaterials with the lead asset on IGT-001. Strong IP that were licensed from Harvard Medical School, and we have potential to expand to larger diseases such as dry AMD or even glaucoma. And we're currently raising, so if you're interested in talking more, asking me any questions later on today, please feel free to come talk to me. Thank you very much for listening.